Hello, science class. Welcome to our first video online. This is something I like to do. I did a little bit last year when we went to our lockdown with the first years last year. Um, and something I like to do to give you guys a better way of maybe accessing material around science right now. Um, and so to start off, it's one of my favorite subjects is when we talk about cells and we talk about microscopes. But before we get started, you notice at the bottom there's some subtitles. Hopefully those work okay. So if you maybe can't have the sound on or you're having a hard time understanding what I'm saying, you have those there at the bottom that you can follow along with as well. And also the beauty of doing videos like these is that you can look back at them. If you know, I will be assigning work around these videos, and if you're really struggling, you can look back at what we've already been talking about. But anyways, without further ado, we're going to talk about cells and microscopes. So one of the things I had you guys answer a question earlier this week was, what are cells? And most of you had a general idea that they had something to do with, somebody said maybe it's what makes up your blood, or somebody said something has to do with DNA. And these are all right to a certain extent. Um, but what we're going to talk about first, before we get to understanding what they are, is kind of where they came from and how we found them. Because if you look at your hand, right, and sneak peek, your hand is made up of cells. You can't really see those, right? So we need these things called microscopes, which you guys have already used. And I'm going to talk a little bit about where those came from and how we found cells. And so in the beginning, we had this guy called Robert Hooke, and you guys will have just watched a video all about these people, so this all shouldn't be new to you, so I'll kind of go through this pretty quickly. We talked about how he used a crude microscope, meaning that it was kind of not what we'd see today, and he witnessed all these little boxes, right? You remember in a piece of cork, um, which is from a plant, as we know, from a cork tree. And he said that these looked like the boxes that monks lived in, which they had called cells at the time. And if we take a look, this is what he saw. This is what Robert Hooke saw in the video where the video was talking about. He took a look under a microscope and looked at a piece of cork. This is what he saw. You notice how these are all like, they all do look like little boxes, right? And I think we've seen these already in the lab. Um, and each one of these is like a cell. If you think of maybe your prison cell, they're all lined up in a row very uniformly. That's what he saw, and that's what we see today, right? And that's where we get that term cells from. So anyways, if we go back, um, we also have the scientist called Anton von Leeuwenhoek, who witnessed, and now he wasn't looking at a piece of cork, he was just looking at pond water, right? If you walk over to maybe a park and take a little pipette of water and you put it on a slide and you look inside of it, you'll see a bunch of little things moving around, and that's what Anton saw. But he didn't really know what they were yet, so he just called them animalcules, which meant tiny animals. He just saw a bunch of little things moving around, so he said, hey, they look like little animals to me. But what Anton did was his contribution to this whole thing was he created better lenses, so, you know, like lenses and glasses maybe. He created a better lens so that you could see the cells better than what Robert Hooke had seen. So what we are going to talk about when we go to study cells, we use these things called light microscopes. Light microscopes, or LMs. You've already used those. You know what those look like. But in a short explanation of what they do is they take whatever's on those slides, those glass slides that you had, the little square rectangular things, it shines a bunch of light through them. That way when you look through the eyepiece, you can see what's on that slide. And what's really good about these is sometimes when you use other kinds of microscopes, which we won't talk about yet, they can kill the specimen. What's really good about light microscopes, though, is they don't kill the specimen. And if you remember when we're talking about the light microscope, when you were rotating that little basin that had the 4 different eyepieces on it, those had different magnifications, right? And those had a magnification of 4 times, 10 times, 100 times. That just means that it's multiplying how big it is. So if this is how big it normally is, multiply that by 4. If you have it on the 4x, multiply it by 10, and maybe multiply it by 100, making it that much bigger, right? Okay. So while I'm going through these, in every video that I make, we're going to have something called a comprehension check. Just making sure, are you paying attention? Are you following along with what I'm saying? And everyone should be able to answer these because all the answers are in the slides or in this video already. So if you don't know right now, you pause the video, go back, and try and find the answer, okay? So the first question, what did Robert Hooke think the little boxes looked like in a piece of cork? And what different magnifications do light microscopes use? Those are all in the slideshow and my video already. So what I want you to do is either write that down on a piece of paper, all of these, and take a picture of that, send it in through Google Classroom, or you put it in Google Doc, and I'll make a place for you guys to submit that on Google Classroom as well. Okay? All right. So like I said, this is what Robert Hooke saw. That's the cork cell, right? Or the cork piece of cork. Those are cork cells. So moving on. Now this is a graphic that explains what we can see with our unaided eye. That means that you can see without having a microscope or maybe like a magnifying glass, what we can see with our unaided eye. We know we can see a chicken egg, right? If I were to go into my kitchen here and get you a chicken egg, you can see that, right? Frog eggs. If you take a piece of pond weed out of the pond, you might see little frog eggs on it without the naked eye, right? Keep going down though, so this is really big at the top here, 10 meters in height, and that's very tall, 1 meter, I'm maybe 2 meters tall, right? We go really small, like micrometers small, we can't see that with our unaided eye, right? We need, what we were talking about is a light microscope, right? Those things we have in the lab. That's what you need to see most plant and animal cells, and we'll talk about what those are in a little while later. And then everything else is still smaller, so we'll talk about what a nucleus is. Bacteria, that stuff that might make you sick, is even smaller than that. And then with all the stuff way down here at the bottom, it's kind of hard to see maybe because of the subtitles, you need something called an electron microscope. So these can't even be seen with a normal light microscope. So viruses and ribosomes and even atoms, like everything's made up of atoms, you can't see that without, even some electron microscopes have a hard time seeing that. But what I'm trying to say is we're going to focus mainly on what can be seen with a light microscope so far, okay? So when we go to look at cells, inside those cells are organelles. And all that these are what help the cell to function. So you think about what you have in your body, you have organs, right? Those organs make you up. They help you to survive, right? Your stomach is an organ, whereas cells have something smaller called organelles. But that we'll talk about those a little while later. So before we move any further, we need to talk about something called cell theory. Cell theory is so important. We've talked about it a little bit already, but basically what it describes what makes something living, right? What makes something living? And with the microscope being made, we were able to study cells, which help us to describe what makes things living, right? And we, again, already in the video, they talked about the guy who made this first ever microscope. Looks kind of like, there's like the little glasses without actually being glasses in the video. So I don't need to talk about that too much. So we talked about Robert Hooke already, a little bit about his life. He was educated at home by his father, who was a clergyman. That was where most of the education came from back in the day. And an assistant to another really famous scientist who we can talk about another time. This is important though. So the Schwann-Schleiden cell theory. These two guys, Matthias Jakob Schleiden, and we have Theodor Schwann. I'm not going to make you guys learn all about their history and life. These two guys kind of helped us to know what cells are today, right? Schleiden was originally a lawyer and then became a botanist. A botanist is people who study plants. And then Schwann was initially a doctor, but then he started to study the notochord of frogs, which is like the nerve cells maybe in their brains. So he started studying the cells of frogs. So we know that frogs are an animal. He was studying animal cells, right? And then if we know that Schleiden he was a botanist, we could probably assume that he was studying the cells of plants, right? So moving forward, and you guys probably already heard this story in the other video, but they go over to another scientist's laboratory and they were talking about a dinner, And they both noticed that there was nuclei, these things in the cell that might have been in both plants and animals, right? And so what they were able to do is basically, it says, thus he extended Schleiden's theory to animals. Schleiden initially had this idea that plants had all these other things inside them, as we can see here. And he assumed that they would find something similar in the animal cells, right? And he was pretty much right. That both plants and animal cells have the same elements. They have a nucleus, which is like the control center of the cell, a fluid content, which is anywhere you don't see where like an organelle. That's what I was talking about, is an organelle, and they must have some kind of wall. And he was kind of wrong here. Not both of them have a wall, as you can kind of see. One kind of looks more round than the other, but they do have a barrier on the outside, which would, if he would have said barrier, I'd give him the points, right? But anyways, moving on. So this is where we got the idea that there's cells in plants and animals, right? But going back to cell theory, and this is so incredibly important, cell theory, after these two guys talked, Schleiden and Schwann, they kind of came up with the idea that all organisms, animals, plants, and that's all they kind of knew about at the time. They didn't really know about bacteria and viruses all at the time. They knew that organisms, so let's talk about trees and bushes and frogs and birds and humans, must be composed of cells, right? If they're alive, they have cells. Then they also said that the cell is the structural unit of life. That means that anything smaller than a cell, they're not defined as being alive. If you take a organelle, so if we go back to this, if you take any one of these little things out of a cell, we're not going to say it's alive because it's not really going to be able to do anything, right? These are the smallest possible living things, right? If it's less than a cell, it's not alive. If it's composed of cells, we can say it's alive. Composed meaning that it's made of cells. And then this is the last point here, point number 3, right? Cells arise, this is a complicated sentence, cells arise by division of pre-existing cells. Spontaneous generation does not exist. And that was explained in the video, right? If you have, and what I can do here is, if you have, I'm going to go ahead and get my pen out real quick, maybe. Oop. Maybe not. That's okay. If you have one cell and you get 2 more cells, those cells didn't just come out of nowhere, right? They had to come from the previous cell. You can't just have them popping up out of nowhere. They have to come from another cell, okay? These 3 will be on probably a test at some point, a quiz at some point. Know these 3 points to the cell theory, okay? All right. So comprehension check. This is pretty easy. I was just talking about it. What are all organisms made of? And what does not exist in cell theory? So both of these answers can be found on the previous slide. What are all organisms made up of? And what does not exist in cell theory? So what we see here is we can see cells come in lots of shapes and sizes, right? These are all different examples of cells. We have a brewer's yeast, which is used to make different kinds of foods and drinks. Those are alive. Those are the cells, right? We have mammalian cells. So mammalian cells are cells in us because we're mammals, right? So these are our cells that we can see, right? Blood cells of frogs, so cells in a frog that help the blood and oxygen move through. But they all look very different from, say, these 2 species of cyanobacteria, which are bacteria, as you can see here. So bacteria, they're not animals, not plants, they're bacteria. They all look very different, right? So there's not one maybe cookie cutter shape, and we'll see that later on, for a cell. Every cell maybe might do something different, right? Our blood cells are different from our brain cells, are different from our skin cells. So moving on again here, we can see that we have 2 main types of cells we're going to cover right now. We have our typical animal cell and our typical plant cell. And what you guys are going to have to do for homework or for additional work today is you guys are going to have to kind of figure out what all these different things are in the cell, right? So if we were to take a skin cell off you, boop, and put it down on a microscope, and you were to zoom in a lot, you'd see all these different we call these organelles, right? and each one has a different job. Okay? Same thing with a plant. If you were to take a leaf of a plant, or we've seen onion cells, right? Onion cells, they all have these different kinds of what we call organelles inside this, what we're going to call right now a cell wall. And I'll go through some of them now, but next week I'll go over all of them. Okay? The most important one right now, this part of a cell, is a plasma membrane. Okay? So it has, and there's a lot of big words here. Phospholipid bilayer, also containing proteins, sugars, cholesterol, acts as a barrier to passage of materials into and out of the cell. Don't worry so much about this first point here. Worry about this second point. So if we go back to our image here of an animal cell, all around here, if you follow my cursor, all around here is this line. It's kind of a grayish, and all out here is this grayish barrier. That is the plasma, or the plasma membrane, right? This is made up so that it can keep out things that don't need to be in the cell, right? It needs to be contained for it to be. If we didn't have our skin, we'd just fall apart. It's a gross thought, right? So this is kind of like the skin of the cell. Okay, keeps out things we don't want in, and it also lets in things that we do want, right? Because it needs to eat, it needs to drink. It's alive, it still needs materials to stay alive, okay? So this is, we can also call it, so plasma membrane, it's also called something called a phospholipid bilayer. That's tough, right? Made up of a phosphate group and 2 fatty acids. This is what it looks like on a chemical formula. Don't worry too much about that. What I really want to look at is this, okay? So if you were to zoom way in to what, I'm going to go way back to this video, to this, oops, sorry. If we were to zoom way into like a spot right along this cross-section, a cross-section is you cut something in half. If you were to zoom way in on that, you would see something that looks like this black and white diagram at the top here, right? Look at all these little bobbles on the top, and then these strings that run down through, and then bobbles at the bottom, right? What's happening here is we have two terms that are really important. We have hydrophobic tails and hydrophilic heads. This word hydro means water. Maybe think of a hydro pump in Pokemon or something like, I don't know, something like that. The water pump. That's what the word hydro means. Philic and phobic. Have you ever heard of a phobia? So maybe you have arachnophobia. That means you have a fear of spiders. So we know that arachnids are spiders. Phobia, phobic, that means fear of. So hydro, water, phobic, fear of. So these tails, which they look like tails, are afraid of water. So what they do then, and then we have hydrophilic, philic meaning I like something, water meaning, or hydro meaning water. These heads like water. So what they're going to do is these heads and tails are going to all arrange themselves. And so say if I have my pen here, and this little bobble here is the head, they're going to arrange themselves so that wherever the water is, the head's going to go there and the tail's going to go the other way, okay? And they do that on either side. All of this to kind of form, if you look at this orange ball down here, and notice that there's a bit of water in here, they're going to form this layer. It's a bilayer because there's two sets. There's a head and a tail, and then there's another head and a tail. They're going to orient themselves so that wherever the water is, whether it's on the outside or the inside, that the head is always facing the water and the tail is always facing away from it. So that is how our cells kind of create this, what we call a plasma membrane or a phospholipid bilayer. So these phospholipid bilayers, the plasma membrane, whichever one you want to call it, their biggest thing, one, is they're protective. So this is a function of the membrane, the phospholipid bilayer on the outside of the cell. They are protective, so they keep things in or out of the cell. So like just as much as you need to keep things out of your body, like bacteria and viruses and dirt and all that gross stuff, you need to keep everything inside, right? I need my heart to stay inside my chest. If it falls out, I'm going to die. It's the same thing with the cell, right? If everything starts falling out of the cell, it's not good. It's not going to be able to function, right? And then cells need to know which cells are good and which cells are bad cells, and that's how phospholipid bilayers they help with that. If there's something that's not supposed to be in your body, like a bacteria or a virus, we need to know which cells are the good cells, right? So that the good cells have little nodes and stuff on their phospholipid bilayers that let that let your immune system know that it's a good cell. And it also, phospholipid bilayers also have specific spots on it where it lets in certain molecules like water and oxygen, things that it needs to live. It lets those molecules in, okay? So that's a function, and this is going to be a part of the work I have you guys do here today, is talking about the function of different organelles, right? And this is the one for the phospholipid bilayer, okay? So another comprehension check. Which portion of the phospholipid is polar, and what are the two parts of the phospholipid? Don't worry about this first question. I meant to change this. I didn't get a chance to. Don't worry about this question here, but you do know what are the two parts of the phospholipid. Okay? So moving on. So this is really quick, and this one I will be able to draw for you on, maybe. So internal membranes, eukaryotic organelles. So I'm going to talk about two things here. Eukaryotic is a term we need to know. So we have two different kinds of cells then, but we're going to focus on this one for now, called eukaryotic. Eukaryotic cells is any cell that has organelles. What's an organelle? An organelle is, if we go back to the animal cell diagram, it's anything inside the cell that's bound by another membrane, right? And what I mean by that is, if we were to, I'm going to pull up a little paintbrush here, right? So if you were to look inside of a cell, whether it's eukaryotic and the other one's called prokaryotic, let's go with the eukaryotic one. If you look inside of the cell, right? So let's say this is a skin cell, right? That's an animal cell because it's a part of an animal, and it's eukaryotic. If you look inside of it, it's going to have all these little, what we call membrane-bound organelles. So say these are just the different structures inside the cell that have a function, right? So let's say this is something called the I can't write with it, mitochondria. I'm going to spell it wrong too. Okay? That's one of an animal cell's membrane-bound organelles. And so we know that this whole cell is, I'm just going to say eukaryotic, because it has these different, and so like we have a big one at the center here called the nucleus. This is where the DNA is going to be found. We have maybe something called a little vacuole. It's a little ball here that carries a bunch of enzymes, right? So we know that this whole cell is eukaryotic because it has these different membrane-bound organelles. And all membrane-bound organelle means is that they're inside of a cell, right? And they have another membrane. Same thing. If it didn't have a membrane, everything inside of it. So if this has a bunch of little reactions and things, and we say it's the powerhouse of the cell. If it didn't have this membrane around it, everything inside of it would fall apart and seep out into the rest of the cell. So it's bound by another membrane. Okay, that's all the word eukaryotic means. Is it has membrane-bound organelles or organs of the cell. All of these little things help the cell run. Okay, so then back to this. That's what all this page here is saying, is that eukaryotic cells or eukaryotic organelles tend to have secondary internal membranes. It separates whatever's happening inside the organelle. So whatever's happening inside this mitochondria, it separates that from what's happening out maybe in this like other area out here, which we call the cytoplasm. Okay. Okay. So last thing, today's work. Make sure you've answered all of the comprehension checks. Okay. I think there's only 3 or 4 of them. Make sure you've written those down in your copy somewhere, and then I'll put a place for you guys to upload a picture of that. Okay, or put it on a Google Doc, answer the questions that way. The other thing I need you guys to do today is pick one of those scientists we talked about. So we have Anton von Leeuwenhoek, Robert Hooke, Schwann, and Schleiden. There's at least 4 of them there. Pick one and describe how they contributed to the discovery of cells, okay? This can be any way you choose. You can make up sentences, you can make up a comment or a comic. You can maybe just draw a simple picture. Maybe you can make a video acting out how they did it. It's up to you, okay? So two things. Answer the comprehension checks and pick one scientist. And then I'll also have a small little other activity for you guys to do in regards to these membrane-bound organelles, right? But that's going to be next Monday for sure. Okay? All right, guys. Thank you so much, and I will see you on Monday.